All right, thanks. Can I start? Yeah. All right, so good morning. So last time I presented the basics of plant cell expansion, and I introduced notions like turgor pressure, osmotic pressure, and cell walls. So in this lecture, I will focus on tissues. So plant cells are formed of tissues, and their morphogenesis is the result of the mechanics of the tissue. So this is a work. So part of the work I will present is done with Ibrahim Chedadi, which I introduced yesterday, and Christophe Godin, both in France. Christophe Godin was my PhD advisor, incidentally, but this work is more recent. Who can tell me what this is? Sponge? Yeah, not too far. Yeah. So this is a drawing made by English polymath Robert Hooke in 1665, and this is one of the first observations of a plant tissue done under a microscope. So in micrographia, Robert Hooke reports a number of experiments he made with microscopes, which were a brand new instruments at that time. And here he cut through the bark of a cork, and he describes structures that you see in the bark. And he describes these structures as cells, okay? And so maybe in lectures or textbooks, you will see sometimes that Robert Hooke credited for the discovery of cells. But this kind of interpretation has been criticized. You have a nice review by Peters in 2024. Indeed, at the time of Hooke, so in Hooke's language, cell has a different meaning than the histological sense that we use now. So the cells were described only 200 years after Hooke. And in fact, Hooke uses the word cell interchangeably with the word whole. And Robert Hooke was indeed a proponent of what's called the fiber doctrine, the doctrine of fibers. And he imagined living materials as networks of fibers arranged in this kind of organization. And so, as a tribute to Robert Hooke, I will here focus on poroelastic aspects. In fact, Robert Hooke saw these pores as structures that we could exchange fluids and water. Okay, so there are two big interpretations of multicellularity in biology, sometimes called theories, although I don't think we can call them theories from a scientific point of view. So around the 19th century, when people started to describe cells, they developed a doctrine called the cell interpretation, or the cell doctrine, which consists in viewing the development of a multicellular organism as the sum of the individual behavior of cells. And so with this reductionist approach, the tacit assumption is that we can describe the entire development of a shape if we understand the behavior of the cell. Now, this kind of idea may make sense in animals where cells move and have active forces, but in plants where things are interconnected and cells just grow and divide, the interpretation of the cell doctrine is a bit more fuzzy. And instead, people have proposed that the cell should be described as a whole, as a single cell progenitor that merely divides and forms compartments. And so in this theory, where morphogenesis emerges as a global property of the tissue, cells are merely subordinate to the overall growth. And in fact, this kind of idea was very well understood by botanists during the 19th century, but with many discoveries surrounding the expression of genes and cell biochemistry, the cell language, the cell-based language has, to a large extent, infused in the discourse of modern plant biology. So instead, in this talk, I will take an organismal approach, and I will focus on continuum-based approaches to model morphogenesis, with the implicit assumption that the cell topology is mostly irrelevant to the growth. This kind of organismal interpretation is based on several experiments. In particular, this is a very famous experiment that has been done by many people. So we take here a stalk. This is an experiment from Kuchera and Niklas. We take a stalk, I don't remember what species it is, and we remove the outer tissue of the stalk, right? And we put the two parts in water so that the imbibition can continue. And we see that the outer tissue shrinks and the inner tissue expands, okay? Which suggests that the shape of the intact stalk is a result of a mechanical equilibrium between two regions, one in tension and one in compression. Now, this has been well described by many people, starting from Stephen Hales and his famous book, Vegetable Statics, in 1727, followed by Wilhelm Hofmeister, who discussed the functional implications of this state of tension within the tissue, in particular in relation with tropism, which will be the topic of my third lecture. Julius von Sachs, another very important botanist, identified then that the cause of this tension was the differential expansion of the cell walls within the tissue. And so he established a clear link between the state of tension of the tissue and the process of growth. In the 30s, with the discovery of auxin, Thiemann and Schneider proposed a hormone-based theory for this tension, proposing that the two regions sense auxin in different ways. And in the 90s, then from the 40s, 30s to the 90s, this kind of problem had kind of disappeared from the literature and fell out of favor. And in the 90s, Kuchera came up with the epidermal growth control theory, suggesting that the shape of the plant is essentially controlled by the epidermis, which is stiffer and controls the expansion. Now, in 2008, Rebecca Van Diver and Alain Gorielli proposed a mathematical model for this mechanical process based on the theory of morphoelasticity, and they studied the space of the morphospace of this growth structure and its instability. Now, this talk is about the growth of tissues, and first we need to define a bit what growth means. So growth is an old problem that has been studied by many people. And an early idea that people have put forth, especially D'Arcy Thompson and people of his time, is the idea that growth is essentially a change in form, okay? So people have studied the problem of growth in terms of specific mathematical functions. For example, D'Arcy Thompson in chapter 17 of his book describes his theory of transformations, where objects are mapped, or species are mapped onto one another via simple mathematical transformation. So the idea here is that growth then is a matter of geometry and a description of the geometry. An early idea to describe this geometry is what's called the theory of allometry, which originates in works by Galileo and anatomists like Cuvier and Dubois, but was mostly synthesized by Julian Huxley and Georges Tessier. Now, allometry at the scale of a single individual relies on the notion that complex morphogenesis requires different parts of a body to grow at different rates. And so the idea is to relate these different rates through power laws, okay? The power law relates a given property of the whole body, such as its size or its mass, to a subregion of this body, okay? Now you can do also this across populations or species. For example, if you say that this is the mass of a human or an adult human, and this is the height, so we can measure, this was done by Adolf Ketley, we can measure the K here is around 2. So the Ketley coefficient here, can you tell me what it is? The body mass index, yeah, which was named like this in the 70s. All right. So to measure the morphogenesis of plants, George Avery in the 30s used this allometry-based measure of the expansion rate. And so in this kind of example of experimental prowess, he isolated, so this is a tobacco leaf, and he cut the tobacco leaf in 70 regions, which he tracked manually. And so for each region, or each group of regions, he could measure the average allotropic exponent, allometric exponent, sorry, which provides a measure of the growth field. Now, this measure has a number of shortcomings. First of all, it's a scalar measure. You can't measure any anisotropic expansion here. And it's also a bit awkward from a dimensional point of view. If you look at the dimension of B, you see that it should have the dimension of y, x to the minus k, where k is a real number. So the interpretation of B is a bit unclear. And in fact, it's mostly irrelevant. Okay, so instead, people in the plant world have started to use tools from continuum kinematics, which are much more robust from a mathematical point of view. In particular, for one-dimensional organs, Ericsson and Sachs came up with what's called the relative elemental growth rate, which is the difference in the velocity between 2 points along the chute divided by their distance, which, as we can demonstrate, is a robust measure of the growth rate. And here on the right, you have interesting tools that I've found in Müller's book that he uses to mark material points along the organ or the leaf using these kind of needles. Okay. Is there any questions so far? Yeah? All right. So this kind of idea can then can be then extended to 2D. This is this has been done by different people. And here you have an example by Wendy Silk and Ralph Erickson, where they measure on a leaf the strain rate and the vorticity, which are concepts from fluid mechanics. So the strain rate is the expansion of the distances, and the vorticity measures the rotation of the tissue. All right. Now, these concepts, which have been rediscovered or discovered by plant scientists, are much older, and they originate in the development of finite strain theory in the 19th century, particularly based on the work of Joseph Lagrange and Augustin Ricochet in France, and George Green, who unfortunately didn't leave us any portrait. You will find some portrait online, but those are not authenticated. So as Abby presented, the main idea of finite strain theory is to relate a reference configuration to a current configuration or actual configuration via a continuous map. So this continuous map can be then differentiated to define the deformation gradient tensor, which characterizes how nearby points move and rotate relative to one another. Now, this is for the geometry. We can also define kinematic quantities, so the spatial and material velocities. Spatial, if you do it with respect to the Eulerian specification or Lagrangian. Can define a rate of deformation tensor, which is the gradient of the velocity, which is really what growth is. It quantifies how nearby points move away from one another. So this is the gradient of the velocity. And if you isolate the symmetric part and the skew symmetric part, you get the strain rate tensor and the spin tensor, which I showed before in the leaf. Okay? All good so far? Now, so far I've described the geometry and the kinematics, and I haven't talked about any mechanics. And we know that animal or plant tissues are under stress. They harbor mechanical stress during their development. So this is an idea that was, for example, proposed by Claude Bernard with the idea of homeostatic stress. And this is an experiment by Dumais and Steele on a sunflower capitulum, where it's a very nice and elegant experiment where they just cut through the capitulum. And what they see is that as you do so, the cut opens, which reveals that prior to cutting, some mechanical stress was holding the meristem together. Okay? And so people have understood that what you observe as a growth deformation is actually the result of a mechanical equilibrium where every part of the tissue has a different reference configuration. And so the entire shape results from this recombination of these regions. Okay? Now, if you want to model the mechanics of a material classically, say an elastic material, this is how you do this. You have the geometry, the deformation gradient. You need a balance of momentum, which we have mentioned in the previous lectures, which expresses the balance of mass, of momentum, so balance of forces. And you need a constitutive relation. Here, for example, I use a hyperelastic constitutive relation with an incompressible material, so you have here a pressure. Okay, but growing materials are not elastic material. Okay? They're open system. And in fact, motivated by the need to model the mechanics of growing materials, people have come up with a different notion, a different definition of what growth is. So from a definition of growth as a change in shape, people have proposed that growth is actually a change in mass, okay, which changes things a little bit. And to model this change in mass, the theory of morphoelasticity has been introduced in the 90s, initially from a paper by Rodriguez, but as Abby mentioned, this kind of multiplicative decomposition was already used in different fields, such as finite plasticity. And the idea is to assume constitutively that the total deformation gradient tensor can be multiplicatively decomposed into an elastic deformation and a growth deformation. Okay. Now, if we do this, we have again the geometry, the gradient tensor, the balance of momentum, and a new hyperelastic constitutive relation, which is expressed as a function with respect to A and not F. Okay, only the elastic component enters the energy. Now, while A obeys balance of momentum and equations that are fairly well described, how to model growth is a whole another kind of problem, because it involves aspects of biology and thermodynamics which are much more difficult to model. And the entire idea of my talk is to try and understand how we can express governing laws for the evolution of the growth tensor. Yeah.
Question. So just to clarify this, is this decomposition an assumption, or you can always do this? And the second is that, why do you mention the intermediate configuration is stress-free? Because if there's some constraint and if you're just growing, there could be stresses, right?
There is stress in the total configuration.
But just out of growth, the first configuration that you write, purely coming from growth.
Stress-free in the sense that G doesn't generate any energy.
I see.
It doesn't enter the stress, the strain energy function. So in that sense, it is stress-free. Now, if you recombine everything, you obtain a stress configuration because you have an elastic deformation. And all of this needs to satisfy compatibility. Independently, these tensors do not have to satisfy compatibility. Okay. Now, I've represented the intermediate configuration as this kind of bunch of regions disconnected from one another, which from a mathematical point of view is a bit fuzzy. You can actually, there is a very nice geometric interpretation for this intermediate configuration, which was developed by Arash Yavari based on differential geometry. Yeah, I won't comment much more on that, but yeah, there is a proper definition for this. Now, the problem is to define, to model.
Excuse me. Yeah, I have. Could you please go back to the kinematic slide, just previous slide? Yeah, just one slide back. Yeah. So here you wrote F equals grad of the deformation map. Yeah. So I understand that like G can be incompatible. So G cannot be a grad of any map.
That's true.
But could F be a grad of, like, is this valid for all cases? Or I mean, if, for the incompatible case or growth, is the composite map F like, is it a gradient or not of a deformation map?
F?
Yeah.
F is a gradient by definition.
Like
It's the
for
gradient
growth
of a continuous map. G is not a gradient.
Yeah. So is it always the case that F is always a grad of a deformation map?
F is always a gradient.
Okay.
Yeah. Because we assume we have a reference configuration, so we can define the map between these. So in this kind of theory. Yeah.
I have a question.
Yeah.
This W over there, is
Yeah.
that energy per unit?
It's
zone
energy
configuration?
per unit intermediate volume. Yeah, it's a good point. Okay, so the problem is to express laws for the growth tensor, okay? Now, in biology, an important problem that has emerged is the problem of patterning. Why do we have 5 fingers? Why do we have 32 teeth in our mouths? I think it's 32. Why zebras have stripes, for example. So one of the most important mathematical contributions in this field, in fact, one of the most important contributions was due to Alan Turing in 1952, who wrote The Chemical Basis of Morphogenesis, one of his most important papers written really at the end of his life. And so in this paper, he proposes that a family of species, more than 2 species, could diffuse and interact, okay? And so you're left with a parabolic system of partial differential equations with here a diffusion term, and here a nonlinear interaction, which at this stage is generic, okay? Now, under some mathematical constraints, which can be derived rather easily, this kind of system can generate heterogeneous patterns in the sense that the uniform, so the trivial solution, which is 0, becomes unstable, and you have this kind of nice pattern that emerge, either stripes or dots or this kind of weird zigzag, okay? And this has generated the field of reaction diffusion equations, which are now part of the mainstream of mathematical biology and widely studied. Based on this idea, people in plants, but not only, have proposed that we could formulate a law for the growth tensor that would include these genes, okay? And this gene pattern. For example, if you have a gene pattern derived from a Turing system or something more complicated, or from gene expression, you can express the growth rate as an explicit function of these genes, okay? If you have a gene expressed at some point, you can say growth is faster here or slower, okay? And this has generated a huge number of models. This is an idea that has been proposed and exploited by people like Rico Cohen in Norwich, and they have done a lot of work around this idea, okay? Now, Alan Turing had a good understanding that this was merely an approximation. If you want to relate the genes to the growth, you have to actually include intermediate steps that include the physics of the deformation and the mechanics. And so he wrote in this paper that in determining the change of state, what one should take into account the changes of position as given by Newton's laws of motion, the stresses as given by the elasticities, taking into account the osmotic pressures, which I will mention in a second, as given from the chemical data, the chemical reactions, the diffusion of the chemical substances. And he writes that this is a problem of formidable mathematical complexity. Now, here I would like to sort of tackle this problem in a very humble way and ask, can we build a theory of growth for plants from first principles where we don't have to specify growth as an explicit function of some gene expression, okay? Is there any questions so far? Yeah? No? Okay, so to do so, we need to go back to plant physiology, which I introduced a bit on Tuesday. And I talked to you about the Locard's model, which I will briefly summarize here. So Locard's model is a model for cylindrical cell expanding longitudinally. And we can define a pressure in the cell and an osmotic pressure, which is proportional to the concentration of osmotics. Now, the water influx allows us to write the balance of mass for the water. And we have another expression for the cell wall yield, which allows us to eliminate the pressure from this system. Okay.
I think can I ask one quick question from
Yeah.
the previous slide?
Yeah.
So in the growth equation that you had, right? Somehow the linear dynamics. So you assume the growth tensor to have linear dynamics. Is there some basis?
Linear dependence in
that G dot is just a linear function of G?
No. Well, so if you yeah, in growth kinematics, the right way to write the growth rate, by analogy with the rate of deformation that I introduced before. So F dot F minus 1 is the gradient of the velocity for the total deformation. So you can actually define the growth rate similarly, G dot G minus 1. Here I just moved G from this side. But G dot G minus 1 would be the correct way to express the rate of growth. So if you, for example, if you have G dot G minus 1, and here you have a constant, well, you get an exponential, which corresponds to what you expect if something is growing with a constant rate. It doesn't have to be isotropic. This can be anything. If this is a scalar, then yeah. Well, no, because you need an identity. Yeah, it will be a isotropic growth. Yeah, but in general, it doesn't have to be. It can be anything. And in fact, authors have introduced notions like polarizers, which are not very well defined quantities that impose a direction of growth, but it's not clear how they relate actually to cell biophysics. Okay. So if we do this, going back to low-cost model, eliminating the pressure, we obtain one single equation for the rate of growth, and we can derive an expression for the pressure. Okay. And here the pressure again is derived as part of the problem, as part of the solution of the problem. Yeah. Okay. So the pressure is expressed as a function of the parameters of the problem. Okay. Yeah.
So here you made an assumption that the pressure is relaxed. The dynamics of the pressure is taken away.
Yes, this is the steady regime I introduced on Tuesday. So yeah, this is the steady growth, equilibrated pressure.
And so this is pressure relaxes fast within a cell, but presumably you'll say something about pressure relaxing across cells.
Well, that's the whole point of it. I won't rely on these equations in the next part of the talk. What I want to define is mostly here the phenomenology and what we want to include in a continuum model. Yeah. Now, so this is for a single cell, but people later wanted to model whole tissues, and so Boudon and colleagues, who were my collaborators when I did my PhD in France, came up with these very nice finite element simulations. So here we have a 3D multicellular tissue. Every cell is represented by a polyhedron. Pressure is constant in every cell. And what they model is the mechanical equilibrium of the structure. They have a growth law for the cell walls, and they run the simulations. Okay? And this allows for having very nice realistic structures from actual cells. This is, I think, an artificial tissue, but you can also use segmentations to generate realistic meshes. And you can have here an explicit link between genes and cell properties, because the biophysics is explicitly integrated in the model. And you can simulate here the development of a young flower in Arabidopsis. Here you have here petals growing, or sepals, you have petals. Anyway. Now, this kind of simulation is very nice, but also very computationally intensive. You have to rely on the finite element method, and you have to care about the quality of your mesh and everything. I'm sure you're familiar with this kind of problem. You have large deformations due to growth, which causes significant problems related to the remeshing, and cell division is also a very tricky problem. Secondly, as I said, they impose a constant pressure in every cell, which, according to what I proposed, what I discussed the other day, is actually a non-physical treatment of the Tucker pressure and requires very strong assumptions to be justified. And the last, and to me most important problem in the spirit of this talk, is that this kind of simulation, even if very accurate, is mathematically intractable. And you can't actually get a lot of analytic insight into this kind of behavior. So instead, I want to focus and develop a continuum mathematical theory. And I will build this theory on a phenomenology that is directly taken from Locard's model and articulated in 3 principles. The first principle is the principle of volume balance, which says that a region that grows by a certain volume V needs to receive a certain amount of water equal to V, or yeah. And this tells us that growing regions, fast-growing regions, are water sinks, okay? Their water potential has to be lower than their neighbors. Now, the second principle is the principle that turgor pressure is a mechanical variable. I do not want to impose the pressure in my model. I want it to be a variable so that we can cover all the possible scenarios where hydraulic may be non-negligible. And the third scenario is that growth results from yield and remodeling of the cell walls under deformation. So here growth is not a function of the genes instructing a certain growth field, but it needs to directly reflect a certain mechanical state of the structure. Okay, is there any questions so far? This is quite important. Yeah.
Slow growth.
Say again?
So the assumption of slow growth, the G tensor that it goes with, so the time scale associated with capital G and capital F, the two might be different, right? How do we handle it actually?
Different than
so
what time scale?
The time scale at which the growth happens will be different from what the elastic deformation time scales are happening.
Yeah, I forgot to mention that. So in morphoelasticity, we always assume that the elastic part is at equilibrium. So the elastic relaxation, which is on the order of the speed of sound, is much, much faster than any biological process you may want to care about. So here, we always assume mechanical equilibrium for the A tensor, but the growth dynamics, in contrast, is slow. So we keep an equation for the G dots. Okay. Okay. So the first part, so I said in my introduction, I was going to talk about a poroelastic description of the plant tissue. So this is where it comes. So poroelasticity is a theory of fluid saturated solids, like sponges or soils. You want to model, for example, sand with water, as we saw last time in Joel's talk. You have to use poroelasticity, or yeah. So poroelasticity is a theory that was described. It has a long history, but it was the modern version is described by was built by Maurice Biot, a Belgian physicist, around the 40s. And there's a number of key steps. So we define. So the key idea is to represent the porous medium as a continuum domain, where every phase of the material, so the matrix that defines the cell walls, is defined, so is everywhere present. So we have the matrix and the water content. They are everywhere present and described by a certain volumetric fraction. So we have the solid fraction, the fluid fraction, and the osmolyte fraction, which describes the osmolytes within the cells, so the sugar. And we assume a saturation hypothesis. We say that they sum to 1. Okay, there's no extra air space or anything. Now, the first step is to write the balance of mass for all constituents, so the solid, the fluid, and the osmolyte, and introduce constitutive laws for the transport. For example, typically you would use D'Arcy's law that you may be familiar with. It's similar to Fourier's law in heat conduction. So it says that water will flow towards a lower water potential. But you can derive this law actually from thermodynamic principles from the second principle. And the last step is to introduce constitutive laws for stresses that reflect both the solid phase, but also the effect of the fluid component. Now, in plants, water flows in different ways. It can flow between cells. So this is what's called symplastic transport via plasmodesmata. but it can also flow in the interspaces between the cells. This is called the apoplastic pathway. So in principle, we should describe both these pathways, but here, for the sake of simplicity, I will just introduce a bulk permeability that accounts for everything. And this is a phenomenological parameter. It doesn't derive from any homogenization. This is a future work. So this is D'Arcy's law. The velocity of the fluid is minus the permeability times the gradient in water potential. Okay. This is actually a relative velocity. It's relative to the solid. Now, this is for the water. Now we also have the growth. Okay, so like in Locker's model, we need now to describe the growth of the solid phase. It turns out that there is no well-accepted growth law for the cell walls, neither at the cell scale nor at the continuum scale. So here we assumed phenomenologically that growth happens when the strain is higher than a certain threshold. Okay, and this is something that has been used by authors and seems to reproduce basic phenomenology quite easily, although there is no first principle explanation for such law, and in fact, it's been criticized a lot. We tried to derive a better law from thermodynamic principles using what's called the Coleman-Knoll procedure, but this didn't work as we were planning, and there's still a lot of work around these problems. Okay, yeah. Say again? Yeah, so in Locker's model, which is 1D, stress and strain are roughly equivalent. But in multiple dimensions, especially in an anisotropic material like a plant tissue, the strain can differ quite a lot from the stress. So yeah. Is there any more questions?
Yeah. Will you say more about what you mean by universally passive and or what the conditions are? Because
so the conditions are that, so I don't remember this, I did this quite a long time ago. You have to start, so you, so yeah, you need the growth to be dissipative, so not so passive. You need the growth flow to maximize entropy production. So, for example,
is there a reason that growth must be only
yes,
dissipative?
the origin of this assumption is that it's the idea that plant cell walls yield passively in response to stress. So they've been compared to plastic flow, which is dissipative by definition. Now, it's not actually clear if it should be dissipative even in plants. Some people have proposed that the expansion of pectins in the cell wall can actually contribute to the growth, although these hypotheses are not super well accepted and there's a lot of blurry areas. Yeah, but the working assumption for us was that the yield of the cell wall should be a passive process. We don't want the activity of the cells to come from the expansion of the cell wall. We want to focus on the hydraulic contribution, and we assume that the activity comes from the chemical energy that the cells use to produce osmotic pressure.
Sort of related to that last statement. So when you try to use the Coleman-Knoll procedure, isn't it also true that you assume there is sort of a destination for the growth itself, that it's growing towards something?
No.
That assumption is not?
No, we don't do this.
Because the free energy that you use to, let's say, write down the evolution,
yeah,
you have to make assumptions about the functional form itself, right?
I'm not sure what you're talking about. Maybe we can discuss more about this. Also, the calculations are a bit far in my mind. Yeah, but no, we don't have to do this kind of assumption. We just write the free energy, and you obtain an expression with some terms times rates. So you have A dot, G dot, and P dot, and you have to assume that every term has to be 0 for the inequality to be satisfied. Anyway, this is a side note.
Excuse me. Yeah. So we're assuming that the growth is driven by the tube group pressure, right? So why do we prefer a strain-based growth law over, let's say, a stress-based?
It's because the cell wall grows as a result of turbo pressure, but the actual element that makes the cell grow is that the cell is deformed. The cell wall is deformed. So if the cell wall is deformed only in one direction, it won't grow isotropically. It will grow in that direction. This is why we need a strain or a stress, which is still, as I said, not clear. But pressure would be a bad assumption. You would only obtain isotropic growth, which wouldn't make sense for a plant. Plants grow highly anisotropically.
Hello. What governs the threshold strain actually?
Sorry.
What are the factors that govern the threshold strain? How do we get a hold of E star?
I don't know. It's not known.
Oh, okay.
It's a phenomenological law, and in fact, what controls this parameter or even this parameter, and what are their link with, for example, the elastic moduli, is not completely known. So you have to take this expression as a phenomenological model. Okay, but it's a good question. Okay, we have now a closed system which combines the morphoelasticity and the poroelasticity. So we have a poro-morphoelastic model where we have a description of the water phase with the flux of water, the water potential, the balance of water mass, and the balance of osmolyte mass. And on the other side, we have, again, the multiplicative decomposition, the balance of momentum, where we have neglected body forces, body loads, and an elastic constitutive equation where here P is not a Lagrange multiplier as before, but results directly from the other components of the model. And finally, we have a growth law. Okay? Is there any question about this? Yeah.
Maybe you said it yesterday. The water potential,
yeah.
Where does it, not yesterday, where does it come from, the water potential? Is psi?
This?
Yes.
Well, it's a definition. You can actually derive it from more fundamental principles, but you can just accept it as if. So this is P, pressure, minus the osmotic pressure, which is R, so theta is the temperature, times the concentration.
Okay, so
it's RTC. Yeah, it's just another notation. I should have written it RTC. I did a copy paste from the paper. That's why. All right.
One question. So I understand that right now K is a constant. It's a bulk permeability rate.
K is a parameter.
But is it possible or is it likely that as the material gets remodeled, the permeability changes?
Oh, definitely. You can, if you want, you can add a law for K dot, or definitely. Here, I will take it as a constant. Now, to illustrate a bit the phenomenology of the model, I will introduce a very simple 1D example. Okay. So this is an infinite domain. Imagine you have a root growing on a hydrated medium, so you have water coming from everywhere with some external permeability, so the permeability with the outside, okay? And we will assume that half of this domain has a certain Young's modulus equal to 1. We can non-dimensionalize everything, so we can define this as a reference Young's modulus. And on the other side, the Young's modulus is lower, okay? So this side of the tissue is softer elastically. And this assumption comes from the idea that that the plant is able to rigidify or soften different parts of the tissue in order to control its shape. Okay? Now, we can reduce the problem, so the whole system I presented before can be reduced to this simple system here that involves the velocity of the domain and the pressure. And this is for steady regime, okay? Which means that, so it's as if you were an ant sitting at the border here and you would see the material flow around you, but flowing in a steady way, okay? nothing moves. Well, everything moves, but in a steady way. Okay. Now this is even if it's a simple system, it's still a nonlinear system. You have V. I don't know why I have a nu here. This is a V. You have a nonlinearity here. So the strategy that I used is to do a regular perturbation. So we assume that the softening is very weak. Okay, this region is nearly as stiff as this region. You just have a little minus eta here. Okay, so you expand all variables in orders of epsilon of eta, and plug this in the original problem and separate every order of the problem and treat each order as a hierarchy of problems. Is this something familiar to you? I guess it is. Yeah. The first order is, so when eta equals 0, everything is constant, okay? And we have here an expression for the pressure, which is equivalent to the lockout pressure that I introduced before. This is the expression of the pressure at steady regime in lockout model, just with a different notation rather easily, and we can obtain closed form expression, closed form asymptotic expression for the pressure, okay? So this is all, let's say, exact, well, albeit asymptotic. And we have this kind of pressure profile. So we see that the pressure is high on the stiff region and low on the soft region, okay? And we can indeed compute the drop in pressure, and we see that we have this denominator 1 plus K, K being the permeability with the outside, showing that the drop will be larger when the water stress increases. When water becomes scarce, the drop in pressure will increase, okay? Scarcity of water increases heterogeneities. Now, because we have this drop in pressure, I forgot to say we assume the osmotic pressure is constant everywhere for parsimony. And because we have then a gradient in, because we have a gradient in pressure, we have a flow of water. So these cells suck water from their neighbors, okay? And by doing non-dimensionalization of the problem, we can also identify a single length that governs the problem, which we call the hydromechanical length, which is the square root of all the parameters of the problem, which are the hydraulic conductivity, the Young's modulus, and the wall synthesis timescale, which is the inverse of the extensibility. Okay, and there's no other length in this problem. Now, there are two interesting things to note. First, one thing is that this length is not specified in the geometry of the problem. So if you assume, for example, that the stiffness of this region and the stiffness of this region is enforced by some genes that may be expressed differentially, we see that the pressure is not directly a reflect of some local genetic control, but you have a non-local effect that emerges. Oh, I forgot this. Yeah, I went too fast. Sorry, I went ahead of myself. Okay, if you plot the growth rate now, okay, so V prime, the divergence of the velocity, we see this interesting discontinuity and this jump that comes from the jump in stiffness. So here, in the stiff region, growth is rather slow, and you have an inhibition of growth near the boundary. And here, in contrast, after the boundary, you have an enhanced growth rate, okay? And then growth stabilizes to a constant value. And this jump is directly a result of the water competition that emerges as part of the hydraulics. Okay, so these cells are softer, they grow, and they have easy access to water from these cells, which are slowly growing, and so they steal the water from these cells and reduce their growth rate. Okay, so this is something very reminiscent of an inhibition region, which is a concept from patterning and reaction diffusion equations, where you have this kind of lateral inhibition dynamics. The second interesting thing to notice is that here we just have D'Arcy's law, right? We have something flowing from some high water potential to a low water potential. And this should be, we should expect this to uniformize everything, to homogenize everything. Okay? If you take a sponge and wait a certain time, water will just go through the sponge and everything will be balanced, right? But here we have a sustained heterogeneity. Do you know why? It's not traveling. This is static. Yeah. You have an idea why we have this heterogeneity? Yeah, because this is not static. We have growth. Growth is a driver of non-uniformity, and it maintains this differential pressure. Okay? If things would stop growing, then everything would become balanced. Yeah. What is stuff? Where? Here, yeah. So that's roughly the inverse of the extensibility. So it governs the anelastic yields of the cell wall. It's the typical relaxation time of the cell wall. Okay.
I have a question about force balance here.
Yeah.
So there is a constant, because this is I should imagine this as a 1D stick in a fluid.
Yeah.
So there is a constant fluid pressure from the outside.
Yeah.
And the pressure inside. So P naught is what? Is the pressure outside?
No, P naught is the P naught from the perturbation. It's the pressure you would have if there was no softening. If the material was uniform with Young's modulus 1 everywhere, you would get P naught. So here P naught is maintained on the stiff side, but then things drop.
But you can still have, but the pressure outside.
We say zero. It's just a reference pressure.
Yes.
So yeah, in everything I say, P would be an excess pressure with respect to the outside. What matters is the difference in pressure, not the actual pressure.
And so the osmolyte concentration, for instance, it would be the same.
The osmolyte concentration would be, no, you would have a higher osmolyte concentration here. Okay. But again, you can define a water potential outside and say this is zero, and then just take it as a reference. There's no flux of osmolytes between the outside and the inside.
Maybe now I have a question about osmolytes in the previous in this model.
Yeah. So in this example, I just get rid of this. This is the general model where we want to maybe model some osmolyte flux because cells transport sugar. This is something relevant, for example, in fruits where there's a lot of sugar being transported. But in this example, I just forget about the osmolytes and I just say pi, the osmotic pressure is just the same everywhere.
Because in the general model, usually during growth, osmolytes are created by the system. No?
Yeah, you have a source.
They are the source.
Yeah. So you have here a passive advection and here an active transport, which we don't specify. Okay. Is there any question regarding this example?
So I just have one. So if I understand correctly, what you're saying is that the growth on the softer region close to the interface is higher, and doesn't matter whether there is a continuous flow of water. Yeah. The growth at the far field in the softer region is always lower, and that's something that's sort of known or it's measurable.
No, this is a highly
idealized
pedagogical example. I don't think it corresponds to anything in reality. You can imagine this to be a model for what happens at the boundary between two regions of different stiffnesses, which happens in meristems where you have fast-growing organs and the rest doesn't grow. And I will say a bit more about this.
Yeah, excuse me. Is there a growth law incorporated in this example, or?
Yeah, the growth law would be here.
Okay, so the one-dimensional strain, basically.
Say again?
The one-dimensional strain, is it?
That's the, yeah, I also assumed linear elasticity for this example. So this is the linear, this is the infinitesimal 1D strain. Okay. So that's the second example, a bit more biology motivated. Let's imagine we have a root here, and so roots grow mostly by the tip. So there's a region of the root called the growing region, which expands at a constant rate. But the key is that this region has a constant length, which means that if we do this kind of experiment, this is called a chemograph. So you take a photo, so you let's say you take the image, you take an image along the route like this, and then you stack this image next to one another, and you get this kind of image where you see here the tip of the root having this kind of linear, well, constant velocity progression. Okay, this is a nice experiment that was done again by Ericsson and Silk in the 80s. So to model this, what we can do is assume that we have an infinite half domain, and we look at the problem from the apex, and we say everything is steady. So we are like we're an ant sitting at the tip of the root, just looking at the material flowing away from us. And we assume that the Young's modulus is higher in the far field, and then is slowly becomes lower next to the tip. Okay, so the tip region grows faster because it is softer. And you can imagine that this is due to some hormonal control where auxin is produced at the tip and then diffuses, which imposes a typical length for the auxin control. Okay, but I don't want to enter into too much biological details. Let's just posit that this is the stiffness. Okay. Now we can again look at the growth rate. We see that there is a finite region that grows. So this is the region. So this is the strain. The ratio between the strain and the strain threshold. So below the dashed line, the strain is below the threshold, so nothing grows. Everything is just elastic. And above the threshold, you have this region where the plant grows. Okay, and the key is that the region, so the region where the domain grows has a finite support, right? So we can integrate and we obtain a finite velocity. Okay, so for any parameter, we have a finite velocity for the root. Okay, so we don't have an exponential expansion as before. Now, if we look at the pressure, yeah. Is there a threshold what? B? Yeah, actually it was K in the previous example. Sorry, I mixed up with the notations. Is there a threshold for V? I think so. I remember there is a maximum velocity. Yeah. I don't remember the expression. The onset of the velocity.
So the tip velocity goes to 0
here. Yeah.
Yes. At what value of B does it go to 0? Is it
at what value? At 0.
Okay.
If you don't have any water coming from outside.
So there is no threshold. So any vascular permeability is sufficient for getting this.
Yeah.
Okay.
Yeah, because if B equals 0, you are right at the dash line. So if you have a slightly higher B, you will always capture a little bit of growth. Okay, now we can look at the pressure. And again, what we see is there is a drop in pressure at the tip of the root. Okay, and so the tip of the root is the region that expands is the blue region here, and here we can represent the threshold in pressure, which is just the threshold in strain times the Young's modulus, which is non-uniform. And what we see is that there is a large-scale gradient in pressure that extends far beyond the growing region, and it is in fact governed by a length scale that is 1 over the square root of the permeability. Okay, so beyond the region that is encoded by the genes to be a soft region, you have a large gradient in pressure, okay? And this is something that is very reminiscent and actually directly related to experiments done by John Boyer in the 80s. And so John Boyer proposed this idea of growth-induced water potential. So in this example, the growth itself is enough to lower the water potential and pump water from the far field. Okay. Is there any question?
You're saying that the tip region is softer.
Yeah.
And as it grows, is it also being modeled that whatever was tip earlier is no more tip? So the Young's modulus there becomes harder or stiffer.
Say again, sorry.
You know, as the root is growing,
yeah,
the tip region is also changing. Whatever is tip currently.
Well, the tip is always the tip.
The tip is always the tip.
The material point that is there sits there, but every point that is near the tip will be moved away from the tip, and it will stiffen as it ages.
Is it like, so your distance is always with respect to the tip?
Yeah.
Although tip itself is changing, but
yeah,
it's always with respect to the tip.
Yeah. S is the arc length from the tip. Okay. Is there any question? Okay. Now these were just 1D examples, and I have showed you a 3D fully nonlinear theory. So as a last example, I want to go back to the epidermal tissue tension hypothesis, which I presented before. And I will model this problem by representing the stem as a morphoelastic cylinder. Here, we need water at some point, so we assume that there is a vascular bundle located at the surface of the stem. Okay. The location of the vascular bundle can vary quite a lot between monocots and dicots. Here, we just assume that the xylem is really located near the epidermis, and so for the sake of simplicity, we just say it's at the outer interface. Okay, it's just a very crude assumption, and we can rewrite all the equations of the system in cylindrical coordinates. And in this example, I will assume that the stem doesn't grow in radius. Okay, we have fibers that maintain the stem at a constant radius, and instead the stem only grows axially. And if we assume that this growth is steady, we can derive a system of ordinary differential equations. Here we have a z dot, which is the rate of expansion. But in this case, it's actually not a dependent variable. Well, it is, but it plays the role of a Lagrange multiplier that we will use to specify boundary conditions at the end caps. So it's not an actual time and space dependent PDE. This is a system of ODEs. Okay. Now, in this case, quite nicely, there's an exact solution for the pressure, which was also derived by Passuria and Boyer in 2003. And the pressure is just a parabolic profile across the stem. Okay, which means that potentially it can become negative, which is probably something that doesn't happen in plants. And if you don't want this to happen, you need this condition that says that the square of the radius needs to be smaller than 4 times the effective vascular pressure here, divided by the elongation rate minus the bulk perfusion. Bulk perfusion plays the role of for example, the apoplasm, which would, bring water to the cells much faster than the symplastic pathway. And this is very interesting because it's an inequality that relates the geometry of the stem with its hydraulics and its kinematics. So there's a relation that relates these 3 things and impose strong constraints on how fast can a stem grow, how wide can a stem grow for a given vascular pressure. Okay. Any question? So now we can look, at the phase portrait of the system. So as I said, I want to model the epidermal tissue tension. I want the epidermis to be in tension and the inside of the tissue to be in compression. So to do so, I will assume that the tissue has a certain capacity to remodel, and this remodeling is governed by a certain timescale tau. Okay, and I will say that this timescale is bigger than the outside, which means that the outside will grow slower than the inside and will be then in a state of tension. So if I look at this in a moderately large, so this is the radius of the stem, in a moderately large stem, what we obtain is this. So this is the longitudinal stress across the stem. You see that the epidermis is in tension and the pith is in compression. Okay, so if you would cut through the stem, you would see these two regions move, like bending away from the axis. Now, for a bigger radius, what we see is something radically different, where the inside is in tension and the outside is in compression. Although the outside has a higher remodeling time. So the outside grows slower, but because the pressure is so large outside, because the gradient in pressure is so large, the outside grows faster. And if we push this even farther, we end up in a region of negative pressure. Okay. So we can also model the full dynamic case where we allow the outside to, so we allow the radius to also grow. This is done numerically. And we can start here, we see an example where we start from an epidermal tension. So the epidermis is in tension and the pith is in compression. And as the system grows, this mechanical equilibrium switches, and because the stem reaches a critical radius at which the perfusion is not sufficient in the middle. Okay. Okay, so of course these are ideal examples. In reality, the plant will grow and will remodel its vasculature itself, so we need to add more complexity to in order to obtain a realistic stem. But this is just an educational example. Okay, so in conclusion, in this talk I have talked about growth, starting from geometry, kinematics, including mechanics, and now an important problem that emerges is how to model growth as a multi-physics problem, which includes hydraulics, genetics, and other components. As a first step to do so, I started from basic physiology of plants and proposed a tissue-wide generalization of the low-cost model, where pressure is treated as a variable. This is an important point. And again, connected to what I said last time about presenting plants as an active matter, what we see is an interesting phenomenon where growth emerges from dynamic and spatially integrated physical process, which create emergent and non-local behaviors, such as lateral inhibition, which I presented today, and growth-induced water potentials. So here we move from a theory where growth is imposed locally by the action of some genes to a theory where genes just affect the physical properties of the tissue, and then growth is an emergent behavior of the entire system. Now, to conclude my talk, I talked about Alan Turing early in this talk and the problem of patterning. So it was the end of his life. Alan Turing proposed this paper on morphogenes, and he actually had a very marked interest in plants, which developed very early on in his life. This is a caricature that was made by Turing's mom, showing Alan Turing, who was then 10 years old, gazing at the daisies while his friends were playing hockey. And so after the publication of his paper, well, around the time where he published this paper, Alan Turing had the idea to apply his theory of morphogenes to the problem of phyllotaxis, so the patterning and arrangement of organs around the stems that creates this kind of very nice spirals here, again on a daisy. Now, in June 1954, Alan Turing is found dead in his house. And so this work never gets published. It gets published in the 90s in the form of fragmented notes, which are particularly hard to read. But fortunately, generations of computer scientists, physicists, and mathematicians have explored, and biologists, of course, have explored the problem of fillet axis and identified the morphogens that Turing had in mind. In particular, they have identified auxin as a key molecule for this process, and the pin transporters, which are membrane transporters that transport auxin and generate this kind of non-uniform patterns. In 2024, so my colleague Ibrahim Shedadi and collaborators from Lyon did this kind of very nice experiment where they looked at the volumetric growth rate, so of every cells in the shoot apical meristem. So these are organs that grow relatively fast compared to the center of the meristem. And so they look at the volume, the change in volume of these cells, not just the surface. And what they see is that these cells located at the boundary of the organ have a negative growth rate, which is very bizarre in plants, since plants do not have any actomyosin that can pull and generate any contraction. Plant cells do not contract. So they propose that this is an evidence that these cells, which are slowly growing, and these cells which are slowly growing, are being deprived of water by the fact that they are sitting very close to a fast-growing region, which necessitates a lot of water to grow. Okay. Yeah. So this is the end of my talk. Thanks.
Regarding this last bit that you just said, the negative growth rate. So presumably, so this is just measured as the time difference of the volume of the cell, so segmented cells.
It would be V dot over V.
Okay.
Yeah, well, in a discrete way, of course.
Yeah. So how should I, given that you said that yes, you don't have active contraction or shrinkage of cells, so these are these cells being compressed by their neighbors?
They don't have necessarily to be compressed in the sense that the cell walls do not have to necessarily, be in compression and buckle, but they are compressed with respect to a previous configuration where they would have more water. So if you have an elastic deformation,
yes,
you take some water from it, you can still be in an elastic deformation, in an elastic tension state.
Sure. Yeah.
But you have less water. So I wouldn't say it's necessarily a compression, but more like
they're experiencing forces from the neighboring cells.
Yeah, but we don't actually know. It could be that the organ, by folding on top of the meristem, is like actually squeezing these cells. We don't know. But it could also be that they just pump the water from these cells, and these cells have to shrink, and the elastic deformation of the cells kind of decreases.
And can one see if there's an impact on the wall? Because I guess the cell wall is still very rigid. So if you take out some amount of water, the shape of the cell the wall doesn't buckle or doesn't go wonky.
It can buckle. I think there are experiments showing that plant cells can actually do this kind of wavy things. I'm not too familiar with them. It has some elasticity.
Yes. Okay. But it's not in this case. You don't see any.
I don't know. I don't think they looked at this scale. Okay. But in principle, again, there is no need necessarily for the wall to be compressed. Yeah, you would need a much higher resolution for this kind of experiment.
Previous shoot growth problem that you showed, where you have this change in the in the phase diagram that you had. In the phase diagram that you had. Yeah. So can you say a little bit more about what's the nonlinearity that's triggering this change in? Where is
this change in state?
Yeah, where is that nonlinearity really coming from? So that you have this change in.
So I don't know if it comes from a nonlinearity. What it comes from is from a competition between hydraulics and growth. Here, the pressure in the center, the hydrostatic pressure, is very low. And the pressure here is very high in comparison because the parabola is very, very heterogeneous. Yeah. So even if this region has less capacity to grow, because it is loaded by so much more pressure, it is still in a state of compression. Okay? Because the radius is too high and the center of the stem is not perfused properly.
But it's not easy to see this in the equations.
It would be from this. So here, the radius has passed the threshold at which,
okay.
Of course, here it's like a soft transition, but the threshold, sorry, too many animations. The threshold here would be this line. Because after this line, you get negative pressure. Okay, so if you approach the inequality too close, you have this inversion of stress.
But for the first transition, there's a dotted line, the compression to tension scenario.
The compression to tension scenario? What do you mean?
The epidermal tension from A to B.
From A to B?
Yeah.
What's the question?
Like, can you show what happens in the equation?
Oh, well, in the equations, you simply have, a small radius. And the radius is small, so the pressure in the middle is still quite large, and the parabola is actually quite shallow. So pressure is roughly uniform. And so if pressure is uniform, the region that grows slowest, that remodels slowest, would be in tension. Okay.
Excuse me. Yeah. So regarding that, like where the pressure is high, could you please move over to phase diagram? Yeah. Here. So you said that like the pressure is high at the edges, basically. Yeah. So won't we expect the epidermis at the edges to grow slower as compared to like the? So I mean, shouldn't we expect 3 regions basically? Like 3 regions, like the epidermis should still grow slowly as compared to the the stalk near the edges, right? Like, so shouldn't there be like 3 regions? Like the center grows slowly, but you said the pressure is high at the edges. Yeah, but shouldn't the epidermis at the edges grow slower compared to the the stalk near to the edges? I mean, if it makes sense.
Everything grows at the same rate. Everything is you have the cylinder remains a cylinder in this deformation. The only thing that changes is how efficiently the 2 regions can relax their tension. Maybe I misunderstood your question. Can you repeat your question? I'm not sure I understood.
It's working. Yeah. So I was wondering, like the epidermis grows like it's slower compared to the the middle portion, right? So even though like the radius is large, so we can expect. So there's like a yellow region, a green region, and a blue region. Like here. So what I'm wondering is, shouldn't the green region grow at a higher rate as compared to the blue region outside? Because yeah,
it does.
Yeah, because the pressure. Yeah, because you said the pressure is high at the edges.
Yeah.
So I mean, shouldn't the epidermis grows slower compared to the?
Sorry.
But the epidermis grows slower compared to the. Yeah. So shouldn't at the edges, shouldn't the epidermis also grow slower compared to like its adjoining? Yeah.
What do you mean by growth? It does grow slower. The remodeling in the epidermis is slower. And yet it is in compression.
Oh, okay.
It's a bit non-intuitive.
Yeah.
You have this kind of effect when you include like poroelastic and pressure effects.
Oh, yeah. Okay. Yeah. I
If
think I
you-
kind of get it. Yeah.
If you imagine a classic
Okay.
morphoelastic material where you impose a fast growth in the middle and slow growth on the edge, you would get just this kind of.
Okay. So if I understand correctly, so the epidermis still grows slower compared, but then the compression tension, it comes from the pressure, basically.
Again?
Like the tension compression comes from the pressure,
Yeah.
not
Pressure
because
is
of the
high.
growth.
So even if the remodeling is slow, the pressure is high.
Oh, okay. Yeah. Got it. Thanks. Yeah.
Yes, I was wondering if people see this transition between tension and compression experimentally. Like this transition.
You mean this transition from here to here? Don't think so.
So biologically, we stay always.
Typically, what you would see is this. In some organs, you actually have this, which is interesting. I think you have this. But it would be interesting to see how you can transition from this state to this state. I don't know if this is a feasible thing to do.
Yeah, thank you for the great talk. So you mentioned the Turing patterns and reaction diffusion systems, but as far as I could tell, in your models, it's not included or it's not in there.
Yeah.
So I guess it's quite complicated already, but can you include reaction diffusion in your?
Well, that's the great thing with a continuum approach. You could very easily, in principle, I'm not saying the numerics will be easy. You could add, you could combine the system with a reaction diffusion system and define on the same domain. Maybe you could assume that the reaction diffusion equilibrates faster than the growth, which is the standard assumption. And then you could simulate, you could relate the pattern with some parameters in the growth model. Now, I don't think there's examples in plants where Turing model specifically is useful or relevant. But you could definitely come up with a different kind of reaction diffusion, like a French flag model or anything. Yeah. Vincent?
I have a question on the general model. So the page with all the equation that is very frightening to me.
Should have animated them.
Yes, no, but it's so for the examples after, you told that there is no, you don't need any, you don't need take osmolytes into account.
Yeah. I just said this is a constant.
So how much can you simplify this model without keeping some of the physics? So you can remove the osmolytes.
Yeah.
Then you take phi naught as a constant. This is the.
You take phi naught as a constant. You remove this, so you say the osmolytes are not part of the model; they are controlled by the cells. You can remove the threshold. This is what I do typically because it's a pain. Other than that, you don't want to simplify the hydraulics too much. You could say, oh, well, pressure is very low.
This one is
the hydraulics is very fast. The pressure is uniform everywhere. But then you end up with a model that loses its essence in a way.
And you always use the balance of water mass in the.
Yeah. In this model, I assume that the cell walls. The volume of the cell walls is negligible compared to the water, which allows to make some simplification. It doesn't have to be the case, but it's just a bit easier.
Thank you.
Yeah.
You are assuming that the two is not cheap.
Yeah. Where, yeah, you could have so here an important assumption is that the topology of the cells at a continuum level is roughly constant through growth. So the cells divide, and so if you would look at the cells, you would not be able to tell where we are in time. So they all look the same, basically. You could have scenarios where cells expand only and don't divide, which is very common in plants. Sometimes cells really just expand, for example, in hypocotyls. And here you would expect changes in W, changes in K, and changes in all the parameters. So a future work would be to relate these bulk parameters, so the energy, the extensibility, the permeability, to actual cell topology and actual parameters at the scale of cells. But this is a difficult work.
No questions?
All right.
In the elastic constitutive equation, the stress T is pk2 stress, is it right?
There again?
In the elastic constitutive equation, the T, the tensor, is it pk2 stress, right?
It's what? It's the Cauchy stress.
Pilo-Kirchhoff stress
No,
tensor 2.
it's the Cauchy stress.
Okay, yeah.
It's the Cauchy stress. It's always, in morphoelasticity, is commonly noted with a T. Maybe it's Gorrielli notation, like AG, Alain Gorrielli.
Okay.
Yeah. In linear elasticity, you would typically note it with a sigma. In finite elasticity, people name it T. So it's just a convention.
Okay, if there are no more questions, we can break for lunch. Let's thank Hadrien.
Thank you.